Principles of Bioproduct Separation Module 4 Cell Disruption Segment 1 with me, Yusuf from Bioengineering Study Program, School of Life Sciences and Technology, Institut Teknologi Bandung. Separation of biomass from a fermentation broth is normally carried out using filtration and centrifugation that had been discussed in the previous modules. In many cases, the desired products are in the broth, such as antibiotics, extracellular enzymes, and many polysaccharides and most amino acids. In some cases, the products of interest are intracellular. They are not excreted into the broth, but they are in the biomass, such as protein produced by genetically manipulated bacteria. Lipids and some antibiotics are also trapped in the biomass. Cell disruption is required to rupture the cell wall in order to release the trapped bioproducts, which is the subject of this module. Disruption of cells can be carried out using chemical methods and mechanical methods, which will be discussed in this module. In this module, we will discuss about first, cell membrane, second, chemical methods, third, mechanical methods commonly used at larger scales, and last but not least, exercises. Cell membrane. We begin by discussing about the physical structure of microbial membranes. We emphasize on gram-negative prokaryotes. Such cells have no nuclei. Their genetic factors are carried out in a single strand of DNA. The best example is E. coli, the host organism for producing most recombinant bioproducts. The basic cell envelope for gram-negative cells as shown in the figure has three layers: outer layer, peptidoglycan, and plasma membrane, whereas gram-positive bacteria has only two layers, peptidoglycan and plasma membrane. The outer layer, about 8 nanometer thick, consists of a polymer containing both protein and lipopolysaccharides. The second thinner layer of peptidoglycan exists in one form or another in virtually all species. Below this second layer is a gap called a periplasmic space, which is also 8 nanometer thick. Enzymes are often located in this gap. Gram-positive prokaryotes, as shown in the figure, are missing the first outer layer but have the second peptidoglycan and periplasmic space. The third membrane layer is known as the plasma membrane or inner membrane, which is common in both gram-positive and gram-negative organisms. It consists largely of phospholipid, but also contains dispersed proteins and metal ions. These lipid molecules have two parts. A hydrophobic or tail often contains two alkyl groups, and hydrophilic part or head often includes a charged group, a zwitterion, or an alcohol. In this membrane, the phospholipid tails aggregate together and the heads are exposed to the water, which results in a lamellar bilayer as shown in the figure. The three layers have different functions. The outer membrane and the peptidoglycan provide mechanical strength. The weaker plasma membrane, the innermost layer, controls the permeability of the cell, including transport of nutrients into the cell's interior and export of metabolites into the surrounding solutions. The cell interior, known as cytoplasm, is an aqueous solution of salts, sugars, amino acids, and biopolymers, including enzymes, RNA, and DNA. In naturally occurring prokaryotes, the proteins are in solution, but in many genetically modified prokaryotes, excess protein is synthesized and precipitated within the cytoplasm. Hence, disruption of the cell wall is often required to isolate the protein of interest. Eukaryotic cells possess true nuclei. For instance, yeast, as shown in the figure, have a membrane surrounding the cell which is similar in structure to the prokaryote membrane. Eukaryote membranes contain steroids, which are absent in prokaryote membranes. In addition, the cells also contain organelles, which has a specialized function. Hence, eukaryotes are structurally more complex than the prokaryotes. For the next segment, we will discuss about chemical methods to rupture cell. Until then, I am Yusuf from Institut Teknologi Bandung.